Fine, fine. So we are talking about nanopore DNA sequencing method, right? Nanopore DNA sequencing method is the third generation sequencing method when it comes to DNA sequencing. The first generation is, yes, the Sanger's method of DNA sequencing, which is also called as chain termination sequencing method or dideoxy DNA sequencing method, dideoxy chain termination sequencing method, like that. So when it comes to nanopore method, it is a single molecule detection method, third generation sequencing methodology, you can say. And of course, it has got a really very broad scope of when it comes to scientific research and of course in diagnostics, right? Now you can call it as cutting edge technology, or you can call it as high throughput method, where you are using the biological nanoscale pores to detect nucleic acid molecules, right? So you are going to sense presence of single molecule, and that sensing is based on the basic molecular nature, size, shape, and the charge of the molecule. So when such charged molecule of specific dimensions passes through a nanopore, 1 nanometer diameter, I'm saying nanopore, then uh, its passing through that pore is going to be studied. How it is studied? It is studied or analyzed with the help of deflection in the electric current. That means you are going to have a membrane. In the membrane, you have these nanopores built in, and the DNA is going to pass through that pore, and the entire assembly is in ionic solution, and a current or a voltage is applied. So deflection in the current is going to tell you about this the about the nucleotide base that is passing nucleotide that is passing through. So depending upon the nitrogen base, the size, shape, and charge is going to change, as it is going to be very specific for each molecule based on its molecular nature, and the functional groups attached will decide its charge. So, though overall negative charge is there on account of phosphate, individual nucleotide is going to have variations based on its functional groups present in it, right? The type of molecular nature it has. So each nucleotide is going to give a unique deflection when it passes through the nanopore. So when we talk about nanopore technology, it is in simple words to say it is the DNA sequencing, where you are going to allow the DNA thread to pass through the nanopore, and as the DNA passes through the pore, it creates a signal, and that can be converted into a readout, and that will tell you about the specific base. So nanopore sequencing technology is also called as strand sequencing, as the entire single DNA strand we are going to allow to pass through, and its analysis with reference to composition is done. Now when I'm saying the nanopore, the nanopore can be of any material. It can be as simple as protein, because the very first nanopore that was utilized, or was used for um, originally in the nanopore methodology, it was not synthetic one, it was biological nanoscopic. Of course, you can have synthetic nanoscopic pore, or you can have biological one. The very first one that was used was Staphylococcus aureus toxin protein, right? Endotoxin that was used, and it is a porous structure. It has got a inner pore structure, and that structure is such that that it is like a tunnel, right? But a very small diameter it has, so that a specific size or dimensions are there, and you can insert that endotoxin protein into lipid bilayer. So can you imagine that Staphylococcus aureus alpha hemolysin, the protein that is secreted by Staphylococcus, this secreted protein is pore forming toxin, so it is inserted into the lipid bilayer, or bilipid membrane, you can say. Of course, that is what actually happens in the host also, right? It gets inserted into bilipid membrane of the host, and as a result, osmotic disruption of cell occurs and cell lysis. So that pore of the hemolysin, staphylococcal hemolysin, if you see, It is alpha HL pore. It is the one which is heptamer having 7 units, and it has about 2.6 nanometer transmembrane channels. Of course, the general structure of protein, what we talk in terms of beta strands and antiparallel beta strands, etc., are there, but the narrow diameter of the pore that is formed, right, which is going to allow flux of osmotic components from the cell to outside and cell dies. That diameter of the pore is enough uh, to work as a nanopore, and initially it was used to allow the passage of single stranded DNA, right? And uh, that is how any protein you can choose select on the basis of its properties, and it can be inserted into the membrane, and that would be working as a nanopore. Now, to see the details about what the nanopore structure is, so when I say nanopore, I'm going to have locally, you are going to what they are made up of and how are they going to work, right? So as I said, nanopores can be biological ones or synthetic ones. When I say synthetic, they can be fabricated also. So exotic carbon material like graphene also can be used to prepare the pores, right? Or you can use biological pore forming proteins like alpha hemolysin, right, which is inserted into lipid bilayer. So either of the two things, that is either biological or synthetic materials, can be used, that can be acting as the nanopores. Now these nanopores, they are going to allow the passage of the nucleic acid strand through it, and as the nucleic acid strand passes through it, gets translocated through the pore. One at a time, a base is going to pass through it, right? And as one base, or which acts like say a block or a bead, it passes down the pore, it is going to show its effect. The effect is in terms of deflection of the current, because you are applying a small voltage to the system which is about 100 millivolts. It is applied across the membrane, and the membrane has got two sides, two chambers, and they are filled with aqueous electrolyte solution, because conductivity is to be there, the voltage applied has to flow through. So ionic solution is there, through which you are going to pass the current, and a continuous voltage is applied, so continuous current is passing through. But as the bead is immersed into it, because of its charges and molecular size, when it is in the pore, the deflection is caused or translocation is caused, and that deflection is measured and a signal is generated. So the current through the pore, that is normally going, and it is deflected or translocated when a bead or the block, or what you call a single nucleotide, passes through it. So that deflection is measured by standard electrophysiologic techniques, right? So uh, how the structure would look like. Here is one example, that is the protein. This is the inner diameter of the protein, and which is going to act as a tunnel, something like the transmembrane proteins, which allow transport of certain solutes across the membrane, right? So similarly here you are going to allow transport of this polynucleotide through it. Now here, the principle of nanopore sequencing, when you talk, a biological nanopore is here shown, that is the protein. It is embedded in the membrane, and a high electric resistance the protein has to have, otherwise it will get denatured. So protein is to be chosen that way, and then it is immersed in a electrolyte solution. So ions may only pass through the nanopore. The size of the diameter of the pore is such that it is hardly 1 nanometer, and specific size molecules only can pass through it. So potential is applied across the membranes, so ionic current is generated, and a continuous current is getting passed through it. So such signal is appearing, and as one particular nucleotide is coming inside, then in the particular region it will cause a deflection in the passing current. That particular zone is referred to as sensing zone. So disruption of the current would occur in the sensing zone, and that will be recorded as signal. All right. So when I say this, now here, uh, you have to go on collecting the readouts of the electric signal, and as the DNA passes through it, you are going to get such signal generated, and that signal is recorded and analyzed. So as I said earlier, the nanopore is generated using a transmembrane protein, for example alpha hemolysin pores that I refer to, that staphylococcus aureus for example, right? Or here it is the schematic diagram that shows the alpha hemolysin pores, which is having covalently bound cyclodextrin and 6N mono cyclodextrin molecule. Of course, I mean, you need not remember the entire structure, but for your understanding, I'm just reading it out. 6 deoxyamino 6N mono pyridyl dithiopropanil cyclodextrin. So this is the chemical nature of it. It is symbolized also here. You can see the symbol for the molecule, and it is going to get arranged in such a fashion that the inner core is going to act as a pore. See, this is the schematic representation for the uh, protein, right? So this is the inner core, which is going to act as a tunnel, is going to allow the passing of the DNA thread to it through it. All right. So this is plus sign, this is minus sign, and conductivity is shown with these signs. So the DNA which is passing through the nanopore will change the ion current, and that will be recorded. So this is really a very fast method, because you have uh, analysis just on the basis of monitoring of the deflection in the electric current. There is no reaction as such taking place. There are no reagents. Only the membrane is there. The pores are built in it, and DNA is allowed to pass through it. So as it passes down, each bead is going to give the signal, and that signal is recorded. So potency of the process is really very high. So human genome project, which went on for more than decades, now it can be completed very fast if you have systems like nanopore methods, right? So as you are, another thing is that as you are analyzing a single molecule at a, that is of nitrogenous base or nucleotide base at a time, the reliability is still less than the other system. Single DNA sequence do get within reach, and read length are already much longer than with other systems. But matlab, in case of Sanger's method, it was hardly 1,000 bases that you can read through using Sanger's method. But here, yes, longer DNA you can analyze with Sanger's method. You can analyze with nanopore method, and this is how it would look like. So, for example, I have sequence using the pore. So this is the lipid bilayer. This is the protein which is immersed into it, right? May it be the protein like as we talked about the alpha hemolysin protein, for example. Similar such protein is immersed here. Now this is the tunnel that allows the DNA to pass through. So this is nothing but the pore created, right? You can see this polymer DNA thread black in color. So when this DNA is inside the pore, it is referred to as blocked pore, and when it is not there, it is referred to as open pore. And this here you can see the deflection in the current. As the DNA enters, you can see what kind of a deflection is there. Initially, it is 100 millivolt that is passed through, right? The current, continuous current that you are seeing. But as you see, this is coming in, there is a drop in the current. Some amount of current is going to be reduced, and that deflection will tell you that DNA is in, and this deflection values or the current deflection will tell you which particular nitrogen base is under processing, right? So to read this diagram, I would say it is the alpha hemolysin pore, which is fixed in a bilayer, lipid bilayer. You can see it in two stages, that is open pore, and this is the block pore. So open pore where DNA is not there, and block pore is the one where DNA is there. Now here, the different ionic current blockages will be generated depending upon which nucleotide is passing through. So one nucleotide at a time is present in the pore, and that will give proportional deflection. That deflection is recorded, and you are going to have such data, electrophasic data, that can be analyzed for presence of the specific nitrogenous base, and that sequence then can be arranged, right? Are you getting it? When you talk about nanopores, there is one more term, solid state nanopore. The biological nanopores have been extensively developed. uh, they have a relative shelf life, their limited reuse potential, and difficulty in engineering them, these are the concerns. So the synthetic nanopores could be the one which is more what you can say promising, right? So when you talk about the solid state nanopores, silicon based nanopores, which are dimensional and have atomic sized membranes, they are used. So in place of the protein, alpha hemolysin protein, that is immersed into lipid bilayer, you have the solid state nanopore systems which are based on silicon. They are also available. Why? What is the importance of these? The importance is against that of biological ones like protein based nanopores. So when I say protein based nanopores, the problem is that extensively they are developed, no doubt, but compare being biological in nature, they have limited shelf life. Then exposure to current voltage may denature it over the repeated use of time. So reuse potential is minimum. Then you cannot engineer them to the degree that you want. So you cannot, you can say that it is not really an ideal route for nanopore sequencing, right? So people have tried to engineer the synthetic materials. For example, you have silicon based membranes, and they are used as the nanopore membranes, right? So fabricated membranes are going to be good in terms of like against high voltage also they can be worked, and they can be repeatedly used, and then they can be integrated with microfluidic devices and so on. And of course precision is going to be good. So a nanopore system, when it comes to solid state materials, then these materials can be constructed and extensively used. So silicon based or glass capillaries, or even graphene monolayer assemblies, molybdenum disulfide, that is MOS2 layers, these materials mm, are chosen because maximum voltage that is what you say can be used during the translocation experiments. So these materials are much more robust than the biological materials like proteins, right? So the advances, when you talk about nanopore, you are going to have the nanopores which are going to be made up of synthetic materials like silicon. So silicon based systems are proposed as material for the application because the theoretical thickness can be narrowed down, a very thin dimensions you can prepare, high voltage I mean it can withstand high voltage, and it can be operated under higher bandwidths, and the resolution also can be good. When it comes to advances in solid state nanopore materials, electron irradiation based technique for silicon membrane nanopore preparation is developed. So to increase the conductance detection, right? Conductance detection, that is the signal detection, uh, can be maximized, and you can reduce the baseline noise. That is, if there are baseline noises, then original signal gets masked and the resolution is less. So silicon based systems, but they are developed, but with advances. What are those advances? The advances says that it is going to have better conductance detection. If you talk about graphene, then graphene monoatomic layer of carbon grid structures. They are really good when it comes to electrical conductivity and malleability, etc. Of course, experimental parameters also it can withstand very well, say pH, temperature, ionic solutions, etc. And unlike silicon, graphene is stable as monolayer system and can exist as very very thin membranes structures. So zero point 0.3 nanometer structure also can be assembled. So graphene is used uh, uh, on silicon chips and used electron beam drilling to create nanopores. So can you just imagine these words, that graphene membrane, on that you have silicon chips, and electron beam drilling is used to create nanopores. The size of the nanopore is 3.3 nanometer in size. So a single stranded DNA and double stranded DNA can easily pass through it. Similarly, uh, two-dimensional materials are proposed for nanopore systems. So Magazine membrane, that is one term, in which you have one atom thick layers of transition metal carbides. Of course, these are the advances. So, uh, like you have in place of protein, you have solid state. In solid state, you have silicon. Better is graphene, and better than graphene or potential substitute to graphene is the one which is one atom thick layer of transition metal carbides. Transition metal carbides मतलब Ti3C2OH2 for example. Ti, Ti, right? Ti3C2OH twice. Nanopore membranes, they are under study and are evaluated for sequencing efficiency. So this is about just introduction on nanopore system for sequencing of DNA. So one base will pass through these pores. Each base passing through the pore will introduce a deviation in the electric current. That deflection in the electric current is noted, which is assigned to the particular base, because each base has its basic molecular nature, size, and charge, which is going to cause proportionate deflection. So the deflection degree of deflection can be assigned to particular base, and that is how, by reading the signals, you can find out the sequence of DNA, that is the base passing through the pore will give you one signal, the second base will give second signal, like that you have series of signals, and by reading the signals you can generate the DNA sequence. Now I'll try to share one video with you. Till then, just give me a second. Yeah, first for Sanger's method and then for nanopore we will see the video. Yeah, can you hear the voice, or else you can just observe it? This is DNA polymerase that is lengthening the DNA strand, and because the DNTP is added, which does not have 3' hydroxyl group. Now further base DNTP, even if it's coming into the series, cannot be added, right? So this is DDNTP, which does not have 3' hydroxyl group. So here you have so many reactions for each molecule are going on, right? And you can see the polymerase is extending the DNA, and at the end you are going to have the fluorescent tag and the DNA fragments generated of different sizes. Shall I repeat? Just watch, right? So fluorescent label is added, right? Dideoxy. So temperature is such that DNA denatures, right? Now polymerase extends it as it comes across dideoxynucleotide. The process terminates. No further extension possible, and the DNA fragment that is generated is of specific size now. That is how you are going to get series of fragments having different lengths and each terminated with specific base. So by reading these fragment lengths and the base at the termination, you can write the sequence. As you can see, the fluorescence of each of them is different, ATGC, right? So can you summarize it? What you observed in the video? And I shall share with a small video for nanopore DNA sequencing method with you. All right, just give me 1 second. I'm sharing it because it would give you a better idea that what than what we discussed till now. Madam, in a lecture, we'll get back to you after 5 minutes. लेक्चर मध्ये आहे. DNA, motor protein, and another protein. Is the nanopore. DNA will enter. The tether is used. DNA enters the nanopore. The second strand is removed. DNA is passing to the nanopore. As it enters the sensitivity zone, a deflection in the current will be recorded. Hundred bases per second is the speed. Sequence strand. These are the right. Now, uh, when I say this, yeah, I want to just share the last slide. Just now you observed the double stranded DNA was there, the tether was there, it removed one of the strands and second strand entered the nanopore. This is what is this technology. Here you are going to use a synthetic sequencing of a synthetic DNA we are taking as an example of. So this is your DNA. ATCG This is the original sequence, right? Now this is converted and then the hybrid it is hybridized, so you have double strand now, and this is allowed to move. That is what you were seeing in the picture, right? So sequencing using a synthetic DNA and optical readout. Each nucleotide in the target DNA is first converted to a longer strand, and that is composed of pairs of two different code units. So code units here are shown in colors, blue and orange, you can say. So these are the code units, and then it is hybridized the hybridize the DNA, and then after hybridizing the converted DNA with molecular beacons that are complementary to the code units. So molecular beacons, that is, you are adding, which are complementary to the DNA strand, and then this is going to hold the DNA, and the DNA is going to enter the nanopore. As it enters the nanopore, the beacons are stripped off, and the DNA strand passes through the nanopore. The sequence of the original DNA is then read by detecting the discrete short-lived photons which burst as its oligo is stripped off. So this is one more advanced in the technique, which was published in Nature Biotechnology 2008. Of course, it is just a schematic representation that you can imagine. You can, I mean, try to understand that how the DNA double-stranded is attached to a system where you are going to use the measurement based on the photon that are bursting or are getting released as DNA enters the nanopore. So this is another method of detection that you are talking about. So this was one method, and the previous method that we talked about is the this one, right? Nanopore DNA sequencing method, where you are going to use the deflection in the current. So current based, and the another one is measurement on the basis of light intensity or photons, measurement of photons, right? So as far as exam is concerned, this particular schematic diagram is enough, and the first slide and first two slides, like what are the pores made up of, what is the amount of charge, how was the pore generated, and what is the deflection happening, what you are going to measure. These four slides are more than enough. This is the example of the protein that is going to be used, but no need to describe it to the details, as I attempted to express it to explain it to you, right? This is the schematic diagram that shows use of the protein as the nanopore and how the deflection is generated. I- If you have any queries, please ask, or else